First of all, let me thank Philippe and all the organizers for their very kind invitation. So, indeed, in the lab, what we do is really try to develop and apply a lot of single imaging assay, a lot of based on single molecule detection, and sometimes also the manipulation of molecules to study like properties at the single cell level, and from the single molecule to the single cell level. And so, just to, in the context of this symposium on seeing is believing, so I wanted to illustrate what we do, an example that been really of great interest to me in the last couple of years, and also I think which is a very nice example of what we can do now with single molecule experiments. And so this project deals with the study of the target search of individual DNA binding proteins and in mammalian cells. So I'll get to it in a second, but I think as maybe a message, and I'll be very happy to discuss it later. I think one field where really imaging is posed to make with a great progress and bring really a lot of really beautiful information is the organization of the nucleus and the genome organization. And although here we're going to look at DNA binding protein moving, I think in general, I think there is really a lot to do there, and hopefully this can be, a first step and maybe give you an idea. I'll be happy to get feedback and during the discussion later. So what I'm going to do first is to just present the people who've been really instrumental for this study. So that was done in close collaboration with Xavier Darzac, my colleague initially at École Normale in Paris. Now we moved to UC Berkeley. And the people, some of them were joint student postdoc, Davide, Lydia, Ignacio, and now Laura. They really played a role at different stage. I must say also that some of the work I'm going to show was done during a stay, the opportunity to do at the Janelia Research Center, and with the help of many people, including Gigi Chen and John McLean, and part of this transcription imaging consortium. Some theory, you saw a little bit of modeling at the end from Olivier Benichou and Raphaël Voiturier at the UPMC. And if we want later more details, I think most of the results I'm going to show are in a paper that's now coming out soon. So to start, I think really to understand the problem, I think the best is to start with this image, which is like a classic game of like searching for Waldo. And as you like probably searching, and some of you may already found Waldo, I think the important thing is try to get a sense of the process you're following. And probably most of you are trying to do the following thing is to search like locally, like something like maybe here. Really go into kind of details, and if you don't find Waldo, like to jump to some other location, search really, exhaustively around this position, and again, if you don't find it, jump at least finally, I think, is here. So you find your target. And this process, which is actually, you can find instances of this type of search process in many cases in the macroscopic world, is called in the literature by statistical physicists like intermittent search process, because you go from very local to like a jump to a more global position far away. Actually is potentially very relevant to not only the macroscopic world but the microscopic world. And this brings us to these problems of DNA binding target search. And the problem from a biochemical or biological point of view is that, of course, many DNA binding proteins involved in transcription, replication, repair, or other processes have to rapidly find a specific target, which is usually fairly small, 5 to 10 base pair within a very long long genome, which gives millions to billions of base pair. And really the question is, what are the mechanisms underlying the search, and even maybe more importantly, controlling its kinetics. And that's really what we wanted to do by having direct imaging approach, just to be able really to get direct access to the motion of these individual searching molecules. So just to bridge the gap with this idea of intermittent search, I would say that in the literature, when people started like thinking about the target search of this DNA binding protein, you could find like different solutions. One is clear, is the 3D diffusion. So it's probably like what would happen in a liquid, where you have like two entities that have to find each other. One is diffusing in this case, and eventually you find, and by diffusing in 3D, you reach your target, and the size of your target is typically like 1 base pair. You have to find it like straight. The second strategy, which is in a sense a little bit opposite and is obviously specific to the fact that you search a target within a linear structure, so, to some extent can be modeled as a linear structure, is that you could start at some point and like diffuse. So we're not talking about processive search, we're talking about diffusive. And if you search, eventually if you stay like constantly bound to your one-dimensional template, you will find your target. The only problem, it takes a lot of time because diffusion is scaled as the distance scale of the square root of time. So it's not very efficient, it's very redundant, so probably that's not what happens. So that brings us to the last one, which is the intermediate strategy, which is when you alternate between this 1D and 3D, which is illustrated in this little diagram. When you go 3D, then you search locally by scanning or sliding, and then if you don't find your target, you go away, which is basically searching for Waldo at the microscopic scale. And why is that better? Because of course, on one side you slow down your motion because you spend time, searching very locally, so your diffusion is reduced. But the great thing is that you increase the size of your target because you don't need to land exactly at the right position. You can land close to it within the sliding lens, and then increase this something called antenna effect. You increase the size of your target, and if you compute the product of these two terms, the rate can be enhanced. So it can be a favorable strategy. So that's good, and that's very much on the paper. So now let's look at it from an experimental point of view. Can we get evidence for that? And just put numbers on all these rates and all these factors, this sliding length, this diffusion coefficient, and so on and so forth. So with Xavier, when we started thinking about this problem a few years ago, we said, okay, let's try to design a good model and a good experimental system to that. And the way we approached that question was to use something which is relatively standard, which is to use a gene array. And the reason for that is that clearly, the key point in the search and the mechanism in this intermediate search strategy is the role of non-specific attachment. In the sense that you have to bind to non-specific target and transiently. And if it's not the right target, not the specific target, then you can unbind and keep going on. So what is key is to be able to distinguish between non-specific binding and specific binding. And obviously, if you take an endogenous transcription factor, for instance, it's going to have a lot of specific target sites that you cannot identify. So it's hard in the motion to unravel specific versus non-specific. So that's why the idea of using a gene array where you know exactly your target sequence, lac repeat or tet repeat, that are located at a specific site, and they are not present anywhere else in the genome in human cells, and that's clearly give you your target locus. And everything else is like a non-specific pool of non-specific sites. And so we use the gene array with like a large number of repetition of lac repeat. Lac repeat are used to visualize the site. And the tet repeat, which are in purple here, it's called transcription factor, is actually a little bit of and it's not maybe so accurate. It's more a DNA binding protein. It's this tet repressor. And so basically that's our searcher, the tet repressor, and that's our target. And so we can now visualize its motion. And to do so, we label it. We just do some kind of brute force approach to some extent, and we inject the searcher and look at it in the context of a single cell at the single molecule level, as you'll see in a second. So first slide, just to show, maybe it's a more controlled experiment, but I think it's an important one. So you have these cells, you know the target site because you have this lac repeat, which is you can transfect with a lac YFP, and then you inject your tet repressor, which is fused to a dye, a red dye, and you see that if you wait a little bit, then you see the spot, and it's the same one. So really, it's just the fact that they bind to the target in the nucleus, and so they can find the locus. So how do they do it? That's the next step, obviously. And to look at it, to address that question, we look at it at the single molecule level. So we inject few of them, and then you see that they move around in the nucleus, that's a nuclear, membrane, which is, schematically depicted. And they move, of course, in 3D, and, could spend a lot of time talking about 3D imaging. Here, everything is 2D, and they move relatively fast. We take about 200 images per second, so that's not real time. It's largely slowed down. And then we can analyze the trajectories. So that's what people like me or in the group doing single molecule tracking experiment they do. You try to look at this trajectory and try to infer some interesting data. So here I'm just trying to give you what we believe is the core message, is that when you look at the first thing we can do is try to analyze how fast these proteins move in the nucleus. And this is a log scale, so you see it's very heterogeneous. It goes from relatively fast, so something which is flying around like almost freely, apparently, to some which are, this kind of part of the histogram, which are this protein, we see the spots are almost immobile. And some have more complicated pattern, but we'll leave that aside for the moment. So the point is, for us, was really to get first an evidence of the role of non-specific binding in the motion, because that's what's... So clearly the good candidate are the protein which are here, these spots, which are not, and I maybe should emphasize it one more time, these spots are not at the target locus. They are like anywhere else in the nucleus, somewhere else in the nucleus. So this is really not binding to the specific site. It's binding to some non-specific site somewhere else. And this is, a fairly large amount of events, about 25% of, all our events. Seems to, at the given time, they seem to be bound to a non-specific site. So, of course, just to check a little bit better that these are really non-specific binding, we did a few experiments, showed two controls. So one of them is we have this doxycycline. If you ever use TET, or reverse TET, that the doxycycline can impair DNA binding in this case. And if you also, we could do another experiment, which consists in incubating the repressor with small oligos which contain the TET operator. So they basically form a complex, and you inject these. And in both cases, you see that you have no attachment to the specific site, and we also think that it affects a non-specific binding. So you do the same type of experiments, and you analyze, and you see that you have no kind of slow event. So really, you seem to abolish the large majority of this non-specific binding. So this is points to some really interaction with non-specific sites. So once we have this kind of things in mind, of course, the next step is to measure the kinetics, because we really wanted to make it somehow in a quantitative description of the process. And so in the way we do it, as like many people who analyze these trajectories, is try to really get from the single trajectories like this one, where you see something moving, boom, stopping for a while, and then it's going to leave and start diffusing again. So we can take tens or even hundreds of trajectories like this, try to analyze. It's kind of a tedious process, but it can be done. And by analyzing this and looking at the phase where they slow down and then they detach again, we think we can get some sort of kinetics of the non-specific attachment. So when you look at this, these events look relatively fast, on the order of 150 milliseconds. But they have also longer events that tends to be in the tail of this distribution. And of course, we have to make sure that they are not, we don't bias due to the bleaching, because when you look at the single dyes, obviously they bleach after a while, so you have a kind of a limited window. So you have to, if you want to look at longer events, you have to overcome that, and the standard and easy way of doing it is try to not do tracking, but some more like time lapse. So we take an image, instead of doing continuously, we do one image, 5 milliseconds, every 100, 500, or 1,000 milliseconds. So when you do that, you get longer events, and then you can pretty much put that all together to get a picture of an entire distribution. of binding times. So that's how we analyze this. So the way to plot it, instead of plotting in terms of histogram, we plot in survival probability, which is the cumulative probability, or defined as a probability to have a binding time longer than time t. So if you do it, you can do it for different set of data set. I'm going, and then you can plot the entire distribution of survival probability. So it's, of course, the shortest events are here, about 10 milliseconds. And so everything in our experiments appear as longer than 10 milliseconds. So you start from 1 and you go down to 0. So there are a lot of, I think, interesting information in this graph that I'd like to comment. The first thing is that you have this, this kind of, it starts and it follows a little bit of this red line, which is the exponential distribution, the Poisson kinetics, with the times 150 milliseconds, the one I showed you before. But as I said, you have this tail of long events, and this seems to stop at a time which is this kind of distribution, this orange, which corresponds to the specific binding. So you go from very fast to kind of long on the order of tens of seconds, which is the specific binding to the target that we can measure in vitro. So it seems like you have like a not, instead of having specific binding versus non-specific, instead of having black and white, you have like kind of more a level of grayscales, and you have like long, broad distribution. So maybe I'll comment that in a little bit more in a second. The first, in terms of quantitative number, what you can get is try to extract the non-specific binding times, which is on the order of 2 seconds. And since the 25% of the bond protein, it corresponds to of the 25% of bond protein, that means the time in 3D, since you alternate between 1D and 3D, is on the order of 3 times that, 6 seconds. Okay, 2 seconds, 6 seconds. So maybe we can make a little bit more sense of these numbers. And for that, I just put like in a table of the value that have been reported. And I think it's interesting to see that, if you look at this non-specific binding in 1D, it's on the order of a second in mammalian, an eukaryotic system. If you go to E. coli, it's much shorter. So I think there is like a huge discrepancy there, maybe for, probably biological, biophysical reason, but it's really remains to be explored why it's so different. And when you look at the kinetics of non-specific association, the 3D, well, it's actually a little bit of the same, on the order of a few seconds for the eukaryotic system. In different systems, some are like TET-R, like fairly exogenous system like ours, but some are more endogenous, like P53, SOX2, and BP1 in yeast. So in all cases, the numbers are differ by a factor 2 to 4, but they're still in the same ballpark compared to what people reported in E. coli for LACI, which was less than millisecond. So really, it seems to have like a different behavior. So I think it's interesting to compare these numbers. Another number we computed, and I did not realize earlier, that if you look at the concentration of base pair of DNA in the nucleus, and that's true whether you're in a bacteria or you're in a mammalian system, just the number scales pretty much with the volume. So the concentration is always on the order of 10 millimolar. But if you move at this speed, which is almost 10 micrometers squared per second, if you're diffusion limited, the reaction should be very fast, 10 microseconds or so, and we find something which is much longer. So clearly, the association to DNA is really inefficient. Of course, when we say 10 millimolar, we know that not all the DNA is accessible. So, you have nucleosome, you have like other many competing proteins which are bound to DNA. So it's obviously not a simple picture, but clearly it's really inefficient. So we're very far for any kind of diffusion limit. So that was for us, it's for me, I think it's in the several orders of magnitude there are actually pretty striking. So if I want to give one more comment on this graph, I think, of course, if you have like this broad distribution, it looks like a relatively clean parallel over several decades. So it's, for physicists, biophysicists, it's always kind of puzzling to see this very broad distribution. I'm not going to elaborate because we're not sure. I think what we're trying to explore is a different like collaborators in terms of modeling is try to see whether we can explain this kind of broad distribution in terms of a hierarchy of binding affinity. And this hierarchy could actually be possibly associated to a variety of binding sequence. You go from random, and that would be a very short association, to 150, to something which is quasi-specific. And so, now if you look at the distribution of sequence in the nucleus, maybe we can really like recover this type of distribution. So that's what we're working on. That's our current hypothesis, but I don't want to make any definitive conclusion there. So in the last, remaining, few minutes of my talk, I just said, since now we describe the mobility of these factors, this DNA binding protein in this model system, we wanted to go a little bit further and say, okay, now we just try really to measure the association rate to the target, because everything I'm talking about, as you, of course, you saw, is really about like measuring and trying to infer association and reaction rate directly at the single cell level in the nuclear context. So to do so, we use the mutant, which is the reverse, the teton, and then in that case, if you add doxycycline, it will actually bind. So if you start from this situation and you add doxycycline, this is very quickly trigger the search reactions, and everything that associates to the target will, in a sense, correspond to protein binding to searching and finding the target. That's exactly what you see here when you add doxycycline. In all these cells in cultures, in U2OS cells, you see this spot getting brighter. So you can get numbers from that, and you can quantify the kinetics at which it's the searcher, the increase of fluorescence at the site. A searcher accumulating at the site. So this is a kind of an observed association rate, but in terms of biochemistry, we had to, of course, correlate that to the concentration of searcher, because the more you are, the faster you're going to find. So this, I think, can be done at the single cell level by FCS when you measure concentration. I think FCS is a nice technique for this and probably underused sometimes. And if you do that, you see that you can define a reaction as a concentration, a rate of reaction as function of concentration, and then you see this very nicely linear rate, and that's actually give you an association rate constant, as we do in vitro experiment, we do here at a single cell level, which is on the order of a little bit less than 10 to the 5. So again, if we want to make sense of these numbers, we can compare to different models. I think for the purpose of this, sake of this talk, I think we can just get one comparison, which is what we expect if you have like a target, diffusion limit, where you have a diffusion coefficient and the size of your target. The size of your target, of course, is not now the size of one sequence, because you have a repetition. You see this, you have a large number of binding sites. Here in this gene array, we have a repetition of number of sites, 200 times 96, so it's really a huge, enormous target in this sense. But so we can have this we can measure it by all kinds of imaging techniques. So it's on the order of 300 nanometer. And if you put all these numbers, you see that the rate you would expect is, again, 5 orders of magnitude larger than what you find. So, of course, again, the fact that you find this 5 orders of magnitude, as I'll make clear in a second, is not a surprise. This is, again, the same 5 orders of magnitude that were missing before, and that correspond to the fact you don't associate well. Maybe I'll conclude on that in a second. So a few more maybe experiments that we did just really to explore the type of reaction rate that we could determine there is we say one experiment on opening this gene array, and we did it by using a lac repressor that now which is called to fuse to an activator. A very potent one in this case, but just for the purpose of the experiment, what it makes is to open this gene array. Instead of having a kind of sphere, you have this kind of very open, large one. You increase the size by several factors, maybe 3 or 4, and you can do that. And our hypothesis was that you make a chromatin more accessible in that case, because of course you can induce the transcription of this gene, which is, you have this reporter gene, which is this blue spot. It's a protein located in the peroxisome, and so we thought, so it should make the site more accessible. So the searcher, if they are blocked by the accessibility of the site, should increase the rate. And you see it changed a little bit, but not much. Most of the time it doesn't change, and maybe a factor 2 sometimes. So it's probably not the accessibility that determine the rate. So the last experiment is the following. We said, okay, maybe something else we can test is the role of the number of target sites. So let me maybe explain the experiment. Is that we used another cell line, where instead of having 200 times 96, so 200 times, this is 200 repetition of 96 very close tetoperiod. We compared the data to another cell line where we have 30 times 7. So, and then we can, okay, let me just show you the result and I'll explain the logic behind that. So in this case, we did the same measurement. So instead of having now almost 20,000 binding sites, 200 times 96, we have 210, 30 times 7. And we measure the rate. And what we see that the rate, just doing the same experiment, is actually not changed much. It changed by a factor 4, but it doesn't scale as a number of binding sites. In comparison, now, if you measure the intensity of this spot at steady state, it changed by a factor 100. So that means that most of at least the occupancy of this binding site is proportional to their number. So it's not really that the rate is not because your site is not accessible, is something else. And obviously, if your search was really 3D, the rate should be proportional to the number of targets. But now it changed only by a factor 4.2. So the last thing is to say why we think it happens like this and what it tells us about the search, is to say that it's an argument that was made actually very beautifully by Johan Elf and in a paper in Bacteria a few years ago, which is when you have like targets which are very close to one another, like this 96 or 7, so close distance which is on the order of 20 base pair, which are a distance which is less than the sliding length, basically the protein, as it moves, it doesn't see multiple targets, it sees one target. Because it doesn't make a difference, it's just sliding, and once it bounds, and then it binds. So that, in a sense, that means that what matters is not the number of binding sites, but the number of inserts, which we call inserts is 200 times 30. And then the ratio of the rates should be the number of inserts, because each insert appears to some extent as one target. And that's somehow like a qualitative or semi-quantitative argument that we have. And the ratio here, then it's about 230, it's about 6 point something, 6.6, which is closer to the number 4.2 that we measured compared to the ratio of number of sites, which is 100. So to summarize and finish, I think the picture is that when this protein search, they come, they bind, but they bind non-specifically somewhere, and the rate is very inefficient, as we saw. So that's what we 10 to the 5, and then they slide, and when they slide, they bind, and that's actually the transfer from sliding to binding is, we think, relatively efficient. And so that's why, that's the best evidence. We don't have direct imaging evidence of the sliding motion because it's a very small scale. But I think this type of measurement on these two cell lines actually points to the sliding motion on DNA as the protein gets to its target. So I think it's time to conclude, and as I stand between you and the lunch break, I think it should not take too much of your time. So it's the main message and from this experiment, and maybe also from single molecule approaches in general, is that really it gives you, I think, sheds a pretty interesting light on the kinetics of biochemical processes, and directly in living cells in the context of an endogenous cellular environment. For the problems of DNA binding protein, I think in our case, but now it's clear from also some other experiments, you have this interaction with non-specific DNA sequences, which is ubiquitous in the nucleus. We see that these off-target residence times are broadly distributed, possibly to a variability in the non-specific sequences. In some other experiments, which are not shown here, we see that this seems to be a fairly general observation that we find. So putting like a little bit of blurring the line between, a little bit of fully specific or fully non-specific binding. And also an important message is this association rate constant, which are very small, and probably not because of chromatin accessibility, but maybe because this protein are intrinsically non-specific. I mean, it's hard to bind to DNA for a reason that needs to be definitely explored, and that's really more, and still I think an open question why it happens like this in the nucleus, and whether it's a way to associate and form complexes in a way which is more efficient. I mean, there are a lot of, we can speculate, but I don't have the answer, obviously, right now. And then the last point, which was the potential route of sliding that is evidence from these last experiments with the two cell lines. So I think with this, I'll stop, and I'll thank you for your time and for your attention.